Welcome to our lesson on cell transport, the regulation of movement of molecules and substances across the plasma membrane of cells. You're looking at an image of a plasma membrane. Remember, the main component of all plasma membranes is a phospholipid bilayer. This membrane surrounds every single living cell on Earth. Now, some cells have, in addition to this membrane, a wall that would be out in this area, but in this image, apparently this is an animal cell, so we don't have a cell wall, we just have a cell membrane. All cells have a plasma membrane or cell membrane, and it is composed primarily of a phospholipid bilayer. There's two things that can be said of all plasma membranes. Number one, it is a fluid mosaic structure. I'll explain that in a moment. And number two, the plasma membrane is selectively permeable. Now, fluid mosaic, what does that mean? Fluid means that these proteins that are integrated within the structure of the membrane, the cholesterol molecules, and all the various components that we see in this image are not in a fixed position. They can move around within the structure of the membrane. So this protein might be sitting right here now, and a few minutes later it might be sitting over here someplace. So it's a fluid structure. It's also a mosaic, which just means that it's a large, complex thing that's made up of lots of small pieces. A mosaic is exactly that, something that's large and complex, but it's made up of many small components. So this is a fluid mosaic membrane. It's also a selectively permeable, or some textbooks will say semi-permeable membrane. Permeable refers to the ability of molecules to move across the membrane from the outside of the cell to the inside, or vice versa, inside to outside. This membrane is selectively permeable, and that means that some small nonpolar molecules like oxygen and carbon dioxide can pass very easily across this phospholipid bilayer. But other substances that are maybe really, really polar, like ions that have a positive or negative charge, or things that are really large, like disaccharides and polysaccharides and proteins, those substances, large molecules and also ions with electrical charge, they have a lot of trouble getting through this phospholipid bilayer. It's very, very difficult for those substances to pass through. So the membrane is permeable to some things, like oxygen. It is impermeable or not permeable to things like sugars and ions. So those molecules that have a hard time getting across the phospholipid bilayer, they can still cross, but they're going to have some special process or some special structure to help them get across. And we're going to learn a lot about how that works. So in this lesson, we're going to be covering pages 105 to 113 in the Campbell Biology in Focus textbook. That's concepts 5.2, 5.3, 5.4, and 5.5. Now, I don't know that I will necessarily discuss all of that in one recording. I'm sure you're hoping not. I may break this up into 2 or 3 recordings. We'll see how time goes. But ultimately, that's what we're going to cover, pages 105 to 113. One of the most central, most important concepts in all of this, to make sure you grasp, is the concept of diffusion. Diffusion is defined as the net overall movement of molecules from an area of high concentration, this would be an area of high concentration, to areas of lower concentration. Pardon my messy arrows. But I'm simply trying to show you that the molecule started out in one concentrated area. We call that an area of high concentration. But then they ended up evenly distributed within the space. So what happens is at some point, the molecules will reach equilibrium where the concentration of molecules is the same throughout the space that they inhabit. But it doesn't start out that way. So as long as there's a high concentration in one area and a lower concentration in another, diffusion will occur. And again, diffusion is the movement of molecules. Could be oxygen, could be water, could be carbon dioxide, could be a lot of things. But it's the movement of molecules from an area of high concentration to an area of low concentration. So one scenario where you have experienced diffusion in your life is if you've ever been sitting in a room, maybe a fairly large, like a classroom, and someone new walks in, an administrator walks into the classroom, or somebody that's not normally in the room, and that person who walks in put on way too much perfume or cologne in the morning. And when they walk in the room, initially only one or two people can smell that, nasal onslaught that's coming from the cologne. But after a few minutes, maybe a couple of rows of students can smell it, and then by 10 or 15 minutes later, the whole classroom just reeks of this cologne. That's diffusion. When the person walked in, there was a highly concentrated cloud of perfume molecules just around that one person. The rest of the room had a much lower concentration of those molecules, and the molecules really have no choice. They move from an area of high concentration to an area of lower concentration. Okay, so when it comes to how living cells are affected by this diffusion process, the easiest mechanism to understand is what is known as simple diffusion. So I want you to imagine a cell in your body. It could be any cell in your body, kidney cell, liver cell. Let's just say it's a cell way down in the middle of your big toe. Okay, that big toe cell is surrounded by a plasma membrane, which remember is mostly a phospholipid bilayer, which is selectively permeable, so some substances can pass through it easily and others cannot. So everything below the membrane, this would be the inside of the cell. Notice it says cytoplasm. And then this would be the outside of the cell, this little tan-colored area. Now, I drew in a blood vessel called a capillary. Capillaries are very, very, very tiny blood vessels. Every cell in your body has a capillary somewhere nearby. Capillaries are so tiny and so numerous, there's millions of them throughout the body. It's probably true to say that every cell in your body has a capillary somewhere close by. And every time your heart beats and blood is pumped through that capillary from your, basically the blood goes from your heart to your lungs, then it goes back to your heart, and then when the heart pumps it again, the blood is pushed out to all the capillaries of the body. And as the blood passes through this capillary, there's going to be a high concentration of oxygen molecules because the blood went to the lungs before it went down to the big toe. So it picked up oxygen in the lungs. We have a high concentration of oxygen now in the blood. We have a much lower concentration of oxygen inside the cell. That's pretty typical because cells are constantly using up oxygen. It's usually a fairly limited supply. So there's going to be what we call a low concentration of oxygen in the cytoplasm, a much higher concentration in the blood, and that turns out to be a very good thing. because remember, molecules have no choice. They have to move from an area of high concentration to an area of lower concentration. So in this case, oxygen will diffuse from the blood across the phospholipid bilayer and into the cell. And that will continue to happen. That flow of oxygen will continue until the concentration of oxygen is the same inside the cell as it is in the blood. And that's probably not ever going to happen, because before that can happen, the heart has beat again and a fresh new supply of blood has already arrived. So you kind of have this, as long as you're alive and breathing and as long as your heart is beating, you're going to have this perpetual state of high concentration of oxygen in the blood, lower concentration of oxygen in the cell. And that's a good thing. That's going to allow diffusion of oxygen into the cell, which keeps the cell alive. It supplies the cell with oxygen. This is called diffusion because we're going from high concentration to low. That's what diffusion is. It's simple diffusion because the oxygen is simply able to pass directly through the phospholipid bilayer. And you will find in a later part of this lesson, it's not always so simple for some molecules. But oxygen is very small and very nonpolar, and so it has no trouble slipping right through the phospholipid bilayer. Now, at the same time this is happening, you have kind of the opposite going on with carbon dioxide, because carbon dioxide, represented here as these little boxes, CO2 is a waste product that is created by your mitochondria when they burn glucose. So if you burn a fuel, which is what mitochondria do, you're going to build up waste. And that waste is carbon dioxide, and we need to get it out of the cell. Well, notice it's a high concentration of carbon dioxide in the cell. There's a much lower concentration of CO2 in the blood. And so what do we have? We have simple diffusion. Carbon dioxide moves from an area of high concentration to an area of lower concentration, passing directly through the phospholipid bilayer. So when you're taking notes on simple diffusion, there's a few basic things to remember. Energy is not required. This is a passive process. Molecules that can cross the membrane by simple diffusion include oxygen, carbon dioxide, and to some extent, water. Now, the one thing that's different about water, unlike these other two, water is a polar molecule, as you know. And it's tiny, but it is polar, and so it's going to have a little bit of a harder time getting across the phospholipid bilayer, but it still can pass. How do these molecules get into or out of the cell? By passing through the phospholipid bilayer, and then there is just a simple description of what we mean by simple diffusion. Okay, this is probably a good time to point out a little bit of a vocab terminology thing that's kind of peculiar. If we are talking about oxygen or carbon dioxide passing across the membrane, like we saw in this picture, then we call it diffusion, simple diffusion. But if the molecule specifically is water, if we are talking about water moving into a cell or water moving out of a cell by diffusion, yes, it's still diffusion, but we don't call it that. We call it osmosis. So here's kind of a special terminology. Osmosis is simply the diffusion of water, specifically water, across a selectively permeable membrane. And so I want to show you a scenario that you'll see a lot of times on test questions. And it confuses students because it involves some terminology that sounds just enough alike to be confusing. hypertonic, hypotonic. I want to make sure you understand what is meant by these terms and how it relates to osmosis. So notice in both of these pictures we have a cell, just a simple circle to represent some kind of a cell, maybe a skin cell, blood cell, doesn't matter. And that cell has been suspended in a liquid, a solution, and there's some, at least in the first beaker, there's something dissolved in this liquid, something dissolved in the water. It says 4% solute. So that could be salt. This could be a 4% saltwater solution, or maybe it's some kind of sugar. So maybe it's some sugar water that has a 4% concentration of sugar. So whenever you have a solution, if you know the percent solute, then you automatically can figure out the percent water. So notice inside the cell, in the cytoplasm, it's 1% solute, 99% water. That's pretty typical, I think, of most cells. The cytoplasm is mostly water, but it does have glucose and various ions like potassium and sodium. So it's not 100% water. It's not pure water. There are solutes dissolved in there. So we'll say it's about 1%. Well, this cell is surrounded by a liquid that has a higher concentration of salt or sugar or something. 4% is higher than 1%. And so we say that this solution out here is hypertonic. Hypertonic means that the percent solute is greater than it is inside the cell. 4% is greater than 1%. On the other picture, notice it's the opposite. This liquid out here is distilled water. It's 100% pure water. There's nothing dissolved in it. So 0% is less than 1%. There's less solute in this beaker water than there is inside the cytoplasm. So we call that type of solution hypotonic. Hypo rhymes with low. So just think low solute. In this case, it's 0. You can't get any lower than that. That means there's a very, very low amount of sugar or salt dissolved in this water. In fact, there's none. Now, what's important here is how does a hypertonic solution like this 4% solution, how does that affect the cell? And then how does a hypotonic solution like this distilled water, how does that affect the cell? And it affects it because water molecules will either diffuse out of the cell or in. Remember, molecules move from an area of high concentration to an area of lower concentration. So in the first beaker, water has no choice but to move out of the cell from 99%, which is a high concentration of water, to the outside of the cell, which is only 96% water. Don't look at the solute, look at the water percent. Water moves from high concentration of water to lower concentration of water. Over in the other beaker, notice it's the opposite because we have 100% water out here. It's only 99% water in the cell. And so the water has no choice but to move from an area of high concentration to an area of lower concentration. Whenever a cell is surrounded by a hypertonic solution, water will move out of the cell and that cell will shrink. It'll literally shrink. The shrinking of a cell due to water loss is called plasmolysis, and that's not a good thing for cells. On the other hand, when you place a cell into an environment that is hypotonic, water will move into the cell, causing it to swell. And if this inward flow of water is strong enough, it'll actually burst the cell if it's an animal cell. Now, plant cells typically don't burst because they have a cell wall. But if this is an animal cell, the only thing it has around it is that flimsy plasma membrane. So if too much water moves in, it will burst. The bursting of a cell due to water pressure is called cytolysis. Okay, and then this is basically just reinforcing the same idea. I'm not going to spend a lot of time on this image, but the pictures in the middle here are kind of cool. These are human red blood cells. And you notice the only one that looks right is the middle one here. That's the normal shape and look of a human red blood cell. The one on the left is shriveled up. It looks like a raisin almost. And that's because this cell has been surrounded by a hypertonic solution, like some really salty water or really, really sugary water. And so water is moving out of the cell, causing it to shrink. In the right-hand picture, we have the opposite. This blood cell was surrounded by a hypotonic solution, like maybe distilled water. And of course, that means water's going to move into the cell, causing it to swell and maybe even burst. But the cell in the middle picture is kind of important to realize what's going on there. Let me get a pen, maybe change colors here. What's happening in this cell, and I'm going to see if I can draw this, is we have the flow of water into the cell, but it is exactly equal to the flow of water out of the cell. That's equilibrium. And the reason that would happen is because this cell has been surrounded by what's called an isotonic solution. So that means that the concentration of salt or sugar or whatever the solute is, the concentration is the same outside of the cell as it is inside. And therefore, the concentration of water is the same on the outside as it is the inside. And so you get this exactly equal pushing of water in and out. Basically, they cancel out. There's no change. And we call that an isotonic situation. The solution around the cell, we would say, is isotonic. Okay, so just to summarize osmosis, remember osmosis is a type of diffusion, so no energy is required. Osmosis is specifically the diffusion of water. The water passes through the phospholipid bilayer, usually. Now, it's kind of an interesting story here with a North Carolina connection. There are structures in some cells called aquaporins, which are specialized water channels that allow for fast passage of water across the plasma membrane, because diffusion is sometimes kind of a slow process, and some cells need to move water in or out rapidly. And so some cells have aquaporins. I'll explain more about that in a minute. That this particular cell structure was discovered in 1992 right here in North Carolina at Duke University. So it's kind of cool. But anyway, osmosis is the diffusion of water across a selectively permeable membrane. So if you need to pause it here and jot down the information, feel free to do that. Okay, I mentioned the North Carolina connection with aquaporins. Aquaporins are special channel proteins allowing water to diffuse rapidly across the plasma membrane, much more rapidly than the water would normally pass through just by simple diffusion. Aquaporins were discovered in 1992 by Dr. Peter Agri. I think I'm pronouncing that correct, at Duke University right here in North Carolina. And he found that not all cells seem to have aquaporins, but he specifically observed them in red blood cells kidney cells and many different types of plant cells. So aquaporins essentially speed up the natural process of osmosis. Speaking of osmosis, whenever I was in college and we would do osmosis labs, some of the lab activities involved this apparatus called a U-tube, and it's called that because it's a piece of glass tubing that's shaped like a U. So a little different meaning for U-tube than what you're perhaps accustomed to. But the tube is filled with liquid, either water or some solution, maybe sugar water in this case. And it has two sides to it, a left side and a right side. And you can see right down the middle, separating the left side of the tube from the right, is a membrane, a selectively permeable membrane. This was some kind of plastic with very, very tiny microscopic holes perforating through the plastic. And it's designed so that it mimics a plasma membrane. It is selectively permeable. And basically what that means is the little tiny holes in the membrane are very small, but water can pass through. Water molecules are small enough to pass through the holes, but if you dissolve sucrose, which is a disaccharide, if you dissolve sucrose sugar in the water, the sucrose molecules, which are represented by these large dots, they're too big to pass through the holes. So you notice that we have sugar water on both sides of the U-tube, but you can tell that the right side is much more sugary. It's a 4% sucrose solution compared to only a 2%. So both sides have sugar, but the right side has twice as much. And the question is, since this side is hypertonic, high concentration of solute, and this side is hypotonic, low concentration of solute, can we predict what will happen to the water, which direction the water will flow in this experimental setup? And that's actually very easy to predict, because if it's 2% sucrose on the left, that means it's 98% water. And remember, water can pass through. On the right-hand side, it's only 96% water, because the sugar concentration was higher, 4%. That means the water concentration is 96. You know the rule. Water molecules have no choice. They have to move from an area of high concentration, 98, to an area of lower concentration, 96. And as water moves through the membrane, the level is going to rise on this side. And as water is lost from this side, the level will drop. And it'll look like that. That'll be the end result. And that movement of water will continue to flow from left to right until the concentration of sugar is the same on both sides of the membrane, or until gravity, at some point gravity kind of starts pulling down on all this water, making it hard for it to, to keep rising up. And so sometimes gravity will eventually stop the flow, but you'll see it. You can visually see a shift in the water levels. And that's a way to visualize osmosis in progress. So I mentioned at the beginning of this lesson that I very likely would break it up into parts, and I see now that that's going to be necessary because we're already just about 30 minutes in, and I still have quite a bit left to discuss. So I've gotten through concept 5.2 and part of 5.3. We've discussed diffusion in general, and then specifically we've discussed simple diffusion as well as osmosis. And so we'll pick up in part 2 of this lesson with a process known as facilitated diffusion. So until next time, hope you have a great day.